Now I would like to give you some practical examples of how we address the 3 R's in our OCD research, which means the replacement, refinement, and reduction. So first we have to address that we cannot replace the OCD mouse model with anything else, such as a mathematical model or cell culture. And to be honest, there's nothing else out there compared to our mice. So we are very secure that we're doing relevant and justified research using the mice. Then we have to make sure that we refine our protocols enough and our research design enough so that we minimize pain and distress to our animals. And then we also need to make sure that we reduce the number of animals to the point when we still have statistically meaningful results, but we don't use any excess animals. Now, many researchers, including ourselves, use what is called a power analysis. This is a statistical method to determine, before you actually do the study, how many animals you will need in each group to get meaningful statistical results among these groups. And this takes into consideration the variation that you see that you might expect within a group and between the groups. Now, how do we address these things specifically in our iCook protocols? When we talk about refinement, reduction, and replacement, we have to be very clear about the objective of our study. So in the case of the protocol for this course, one of the objectives was to expose you as a student to animal research, real animal research using real mice. So that was one of the objectives, and that's why replacement was not an option. Now, in that process, we also need to show that we've done a careful peer-reviewed literature review using appropriate keywords to find the literature related to our research. We also have to justify why we use animals. And so here we go into the fact that using human subjects will be inappropriate, that computer modeling or cell cultures are really not available for the scientific objectives that we are looking for. And then that we have actually a model, an OCD mouse model that has face and predictive validity, and we'll talk about that a little bit later on. And that these mice are very valuable, and that what we're planning to do with them is justified because of what we think we can gain scientifically from these mice. So as a result, we have to be very clear about the objectives of our study. And then the third part that we need to address is, why are mice the choice animal for this research? What is the species appropriateness? And so we describe why mice are good to do research with, and I've already told you a little bit about that previously, and how important our OCD model is, and why it's justified to use these mice to do our research. Now, generally, in order to adequately address refinement, reduction, and replacement criteria, it is the researcher's responsibility to be up to date with the peer-reviewed literature and to know what research in the field has been done and what research has not yet been done. And the researcher needs to evaluate whether the methodology or the research design is scientifically justified using previously published research, and whether that research design exposes the animals to minimal amounts of pain and distress. And also that there are no other methodological approaches available that address the scientific questions that do not need the use of animals. And generally, researchers consider vertebrate animals to be maybe more valuable, or they have more cognition or things like that compared to invertebrate animals. So sometimes researchers are asked whether using an invertebrate animal model is possible instead of vertebrate animals. Now, in our case, that's not something we need to consider because our OCD mice are unique. They have high scientific potential and they're great to work with.